Hello, I'm Charles Evevold, and today I'm going to give you an introduction and context to my poster entitled Metabolic Control of Inflammatory Cell Death. So human disease states illustrate the importance of cell death, resistance, or overactivation in pathology. And we can think of this on either side of the spectrum, whereby cells that have acquired resistance to cell death programs might incur pathology in situations such as autoimmunity, cancer, or aging. And on the other side of overactivation of cell death pathways, we can get pathology in disseminated systemic infections such as sepsis, autoinflammatory disorders, neurodegenerative diseases, as well as ischemic injury. Programs of cell death are not all created equally, and some can be termed as immunosuppressive or inflammatory. And here I'm showing you the cell death fate known as apoptosis as an example of an immunosuppressive cell death program that proceeds through apoptosome oligomerization, executioner caspase activation such as caspase 3 and 7, and ultimately resulting in membrane blebing of the cell and exposure of phosphatidylserine such that tissue phagocytes will ingest these bags of DAMPs before that they can be recognized as inflammatory. Now, on the other side, on the highly inflammatory side of cell death programs is the program known as pyroptosis. And this will be the subject of the rest of my poster. And so here, this proceeds through inflammasome oligomerization, activation of another set of caspase species known as inflammatory caspases, such as caspase 1 and 11. And this is a highly lytic and inflammatory program as it proceeds through perforation of the plasma membrane and subsequent plasma membrane rupture that ultimately results in IL-1 cytokine secretion and inflammation. And so the main subject of my subsequent poster is this question, is there novel regulation in the inflammasome pathway and specifically in dictating gastromin D pore formation? And so we asked the question, is there regulation between cleavage of gastromin D and functional pore formation at the plasma membrane? Now, to get at this question, we turned to synthetic biology paired to whole genome-wide CRISPR-Cas9 screening, whereby we took immortalized bone marrow-derived macrophages derived from macrophages from a Cas9 transgenic mouse, and it engineered them to express TET3G transactivator protein, as well as gastromin D variants fused to fluorescent proteins, in this case, BFP. And we focused in on the N-terminal fragment of gastromin D that can form pores within the plasma membrane, as well as the latent full-length form that should not form pores. And here I'm just showing you that if you dox-induce this N-terminus, we can see enrichment of this fusion BFP tag at the plasma membrane, and these cells that have induced N-terminal BFP convert to membrane permeability over time. Whereas if we dox-induce this full-length latent form of gastromin D, you see that it's diffusely cytosolic, and these cells don't convert to membrane permeability. And so then these cells were amenable to a CRISPR survivor screen, whereby we were hoping that some knockout of some positive regulator of pore formation would turn a cell that looks PI positive and BFP positive to a cell that just looks BFP positive that we could sort. And so after we've sorted these BFP positive, PI negative cells, we did a sgRNA enrichment analysis on these survivor cells compared to a whole input sample. And here I'm showing you the results of this analysis as a volcano plot with significance on the y-axis and log fold change on the x-axis. And I've highlighted the right side of this graph as potential positive regulators of gastromin D pore formation, in that if you knock out these genes, you are more likely to be a survivor cell or have deficiency in membrane permeability, but still induce this normally toxic pore forming domain. And I've highlighted several of these members, and these all actually belong to the same biochemical complex. And so if we do gene ontology bioinformatical analysis of our ranked hit list, we also see functional enrichment for vesicular trafficking proteins, as well as mTOR signaling proteins. Again, I mentioned that the 7 highlighted hits on the volcano plot are actually all part of this one biochemical complex known as the RAG-regulator complex. And the known function of regulator is to relay amino acid availability to mTORC1. And so this, the relationship between regulator RAG, mTORC1, and gastromin D pore formation is the subject of subsequent data that I presented in my poster. And I hope that in the live session, I'm able to present and answer any questions you might have. Thank you for your attention.